for joining today. My name is Tim. I'm a software development consultant from Netlight and full stack developer. And I had the pleasure to work on this exciting project together with my colleague Matthias from Bayer. And we hope to share some exciting insights today for all of you.
Yeah, thanks for having us today. Before we get started to dive into how we explore high-dimensional data in early drug discovery with the technology called cell painting, let me briefly start with prompting you to think about what's the amount of data that you think you've created with digital photography since you use digital photography. So I did this for myself and kind of approximated it with this little equation. So I take maybe on average 2 photos a day. It has grown significantly since I have kids. Since about 365 days per year, since 22 years, and say obviously that changed over time, but roughly 3 megabytes of data per image. So if you do the math, then you get about 48 gigs of images that I've created. And I can say, well, at least until the recent developments of image search and cool stuff like that, it's quite a challenge to handle that if you're asked to, I don't know, find a nice image of your mom for their 60th birthday or something like this. Now, compare this with the context where we are working, which is the high content analysis with fluorescence microscopy. So what you see here is one of the microscopes. And these microscopes process plates like this one. So this is a real plate. Their size is standard. And on this plate are 1,536 little wells. They are probably a bit hard to see from your sense, and each well contains a little experiment. So, and I'll get to the details of these experiments later. But if you do the math now, the microscope takes 4 photographs of this 3D well of each of the 1,536 wells. And the microscope can process about 50 plates per day if it runs for 24 hours, and each image, since they are high-resolution images, has about 38 megabytes, so about 10 times the size. So this, if you compute this, makes about 11 terabytes per data, but not in the lifetime of the microscope, but actually on a single day, and our labs have several of those microscopes for fun. So the challenge we are talking about today is how do we make sense of these images?
So onto the content of our talk today. We're going to give you a brief introduction to the process behind early drug discovery, and also in particular cell painting. That's the area where we're navigating. Also for some technical background, we're going to talk or show you our system architecture, and then dive right into 3 different challenges we had with visualizing data, for once in a table, on a scatter plot, and in a heat map, all with the intention to give you insights that you can abstract into your use cases that might be maybe outside of the medtech sector even. And all that to lead to answer the question, how do you visualize millions of data points? Let's dive right in. So the early drug discovery process you can see here is like trying to find this needle in a haystack of a multi-dimensional haystack. And so just to give you a feeling of how big this haystack is, we're looking at 10 to the power of 16 to 10 to the power of 20 possible molecules that you can explore. That is based on how many molecules humans could consume orally, also that are dissolvable in fat. And that explains why the timeline of starting from target and ID validation to clinical trials to time to market can be up to 10 years. The space where we navigate in this?
Yeah. So our application is in the early drug discovery stage, as the title of our presentation already says. And that consists of steps where you try to first identify a target, so oftentimes a protein that has a role in the disease you're looking at. Then you want to validate it. Once you have validated the target, you want to find compounds that actually bind to that target and modify it in a desired way, and then these are optimized. And the technology we are looking at today is called cell painting, can be used in all of these steps depending on how you design your experiments. So how do cell painting experiments look like? As I said, we have these wells, and we grow cells in each well of a particular type of interest. So for instance, cancer cells. And then in each well, we put a different compound at a certain concentration to see how do the cells react basically to this compound in this concentration. Now, how do we see how the cells react? Well, we basically take the photographs, as we saw before in the microscope, and the cells are treated with different stains that contain fluorescences that bind to different compartments of the cell, which makes these very generic parts of a cell, like the nucleus, for instance, or the mitochondria, visible on the photograph in a different color, and that's why it's called cell painting. And now what we do is we actually take these images and create a numeric representation of the images that represents what's actually happening to the cells in a well. So how does their morphology change? That's why you call these representations morphological fingerprints. And so we have one fingerprint for each well, which we call well profiles. But then obviously there's a lot of noise in these images. So in order to get rid of those noise, we repeat the experiments across several wells, and then basically in post-processing, we generate what we call a consensus profile of each condition, basically compound at a certain concentration, and then analyze those conditions to actually find new drugs. Right? Now, when we started to implement this cell painting process at Bayer, here you see again the general workflow. It started like many research projects. So basically, we had a few of bioinformaticians who wrote the first analysis pipelines. There were Python scripts lying around on different computers or servers if your local devices were too small. CSV files were exchanged, sent back and forth. There was a lot of actually emailing, SharePoint sharing, all this kind of stuff to actually generate the insights we were talking about. As you can see, that process doesn't really scale, but as we said, it's a very promising technology that can actually capture a lot of interesting information in early drug discovery. So we were wondering, how can we actually scale cell painting at Bayer?
Yeah, I mentioned the 10-year timeline. You can imagine that every little cycle shortage generation, like making these cycles shorter, can generate a lot of value in the long term. And so we asked ourselves the question, how can we bring that long, manual, tedious process into an integrated end-to-end workflow? And this is how our application, Phenomorph, was born. That's what Matthias and I have been working on together since March last year. And we're going to guide you through the different steps. Here, in a bit of a fast forward, you see on the first image on the left-hand side how images are imported into our platform from the raw microscopic data. Then you see an Airflow UI, but also our platform allows to orchestrate, see the status of Airflow pipelines. And then in a continuous process, it might be that, for example, on the head of the microscopic camera is a bit of dust, or through outside factors like temperature or humidity, some of the images are just invalid. So to exclude all of these invalid data that doesn't meet our standards, we have also a QC step. You see here that we always put a high focus on throughput, so it doesn't matter whether you process one plate or 500 plates at once, all with the intention of reducing these cycle times and making researchers able to do one action on all of the plates at once. And then in the final step, and that's also where we're going to put a focus today, the analysis part and the visualization, where we build a dynamic dashboard-like application that you can, where you can explore your data in real time, you can configure groups of data of interest, and you can go with findings into the next steps of actually finding potential drug candidates. But before we look into the more visual parts, let's look at our system architecture to give you an idea of the whole flow. So on the left-hand side, you see that scientists in a lab in Wuppertal create images. We've seen that on the microscopes. These are stored on-prem. It's a lot of data we've learned, so we do the heavy lifting in AWS. Basically, we have specialized components for each need. We use Batch for the heavy compute, QGen for vector storage, DocumentDB for storing the metadata, and all of the orchestration happens via Airflow that then talks via API with the AWS Batch. The area where we're going to look today is more on the bottom side, our API layer. We put a strong focus here on API first, principles, so that, for example, our API is usable by, on the one hand side, the scientists in the lab that use our visual front-end application, but also we still wanted to enable the old original flows via, for example, Jupyter Notebook by a Python package that we provide. So that allows to explore the data in a way that may be exceeding functionality that we've already built in our dashboard app. And now let's dive right in. You see here a little screen recording we've created on a sample data set. All what you see here is usually possible with up to 5 million data points, but for this talk we couldn't show you original data, of course. So you can see that you can navigate to data points. You see molecular structures. You can get information in real time and also highlight different metadata in the X and Y coordinate system. You can also color the values by, for example, concentration or by control type groups. And as I've explained, we built this with the intention to be maximum adaptable for the use case. So we allowed our researchers to look at different widgets, but also bring in the widgets that they might want. So here you see a dose response curve. But if I don't like this one, I can also look at a histogram and, for example, color the histogram by the same control type that I have in the scatter plot to create some kind of correlation between widgets. Then also another widget that we're going to see today is a table. Again, a high focus on performance. It will render up to 1 million data points performantly, and you can highlight and order by concentration visually. And last but not least, I'm trying to keep up with the video, a heat map where you can see up to 5,000 times 5,000 images in correlation. So we generate cosine similarities, here highlighted by the tone in bright yellow or dark red to show how similar are these molecules.
And now let's have a deeper look into how we actually built this, which is probably more interesting for most of you. So we thought, let's first think about this table. You saw there is a lot of information about these profiles, right? We have the combination of basically this spatial information we create from the representation of compounds as consensus profiles, as we learned, and some metadata. So basically what's actually the compound, right? What's in there, in what concentration, and so on and so forth. So our default approach of implementing the system was to use GraphQL with the schema first approach, and as we heard today, data contracts in the earlier talk, data contracts are important, so that's our way of creating contracts very early for back-end to front-end communication by specifying a schema type, like you see here, for example, a data set, right, which has some metadata, like a name and a description and so on and so forth, and then this important part, the consensus profiles, which basically is a page of profile information. And so with this schema being defined, we can actually generate types in our front end with TypeScript automatically, and then develop the front end against those types, and at the same time develop the back end against the same types. So basically by resolving the fields in the schema. The stack in the back end we use is Fast API as a server, then we use Ariadne, which is a Python package with schema first approach, and then since our metadata store is DocumentDB, we use Beanie ODM, and we'll find those. So to give you an example of how our kind of default approach works with paginated queries, the key here is that you have to decouple actually counting. Because a page default model has always a total count, and then you have some fraction of that total count as page data in your request responses, and you want to decouple those so basically you can do the count that matches your filter criteria separate from fetching the data. And the way we do this is that in Python actually we just, the first level of resolution of this consensus profile fields here, what you do is it just passes on down the tree of resolving this GraphQL query the information of what type of document is actually requested, with which filter criteria, and where in the page represented by a limit and offset do you want to get the data from. And now there's no database interaction yet, but what we can actually do is we can, in resolving the fields of a page type, we kind of attach two different Python functions to the different fields. That's what you see here with this set field To total count, we have a function called resolve total count. And to data, we have a function called resolve page data. And if you look into those, basically, we now utilize this abstract object that was passed on down the tree, the resolution tree, this page query that just contains the document type we can use for the database query, and the filter, which is then pushed down to the database for counting, and then basically offset and limit are passed with it to actually fetch just some fraction of data. So by that, we can actually query pages of data for displaying in the table.
So now that we've learned how to overcome the challenge of bringing the data into the browser, let's talk about how we render it. So conventional DOM tables, like you might have seen in HTML, are limited. So even on a good hardware like this MacBook Pro, you will freeze the browser with 50,000 rows. Nothing will happen anymore, and you can basically close Chrome and try again. So how did we solve our requirement of millions? We wanted custom filtering, sorting functions, dragging and dropping columns, so it was not an option to go with this classical approach. The keyword here is DOM virtualization, and we we found this library here. It's called AGGrid, and it makes sense. It makes use of a technology that basically replaces elements in the DOM instead of rendering them all at once. And that is highlighted here in this little animation. So you can see in the bottom where I inspected the page that the elements are not scrolled down, but they are in fact replaced, and their position in the DOM or the position in the view is changed, but not actually, they're all rendered at once. And that allows to easily render millions of data points without freezing the browser. This is optimized for this purpose, and yes, it comes with a price tag of 1,000 per developer, but I would definitely recommend it.
Now, well, we can't use pagination really for this part of the app, which is the scatter plot, where you actually want to have a look at the data all at once. And without going through the whole video again, fetching all the data all at once actually brought us to a different bottleneck that comes with GraphQL, and that's the validation of each field with several validator functions. So what you see here on the right is that that's the data for a single point, a single profile, and they have a lot of scores that we compute, basically depending on this quality control and all kinds of things. So we have quite a deeply nested large object structure, and we want to have all those fields available for coloring and sizing and so on and so forth. So, but then we realized if we do that for too large of page size or all data sets, then just the validation of the data would take too long, because we have 3 function calls for every single field that we transfer. So what we did is we actually utilized our implementations, but just changed the interface away from GraphQL for selected queries, but basically implementing fast API endpoints directly that would return the exact same type that we have defined in GraphQL, but with no validation now, just to be faster in delivering the data. So we can still utilize our GraphQL schema for as the contract between front end and back end, but we have a dedicated interface for some types of the schema that are sent without validation from the back end. And that way we were actually able to I think reduce the loading times of some data sets by a factor of 12.
So let's talk again about how we bring all that into the browser now, because scatterplot, same principle as with the table. We looked around, and some of you might know D3 as a library for the geometry, if you will, behind different types of visualization. You will see lots of examples provided, but all the examples are written in canvas and SVG elements. So if you render a scatter plot with 5 million points, that would result in, you see down here, different circle elements that all need to be rendered by the browser. So how did we overcome this challenge? The buzzword here is basically using a game engine or a 3D engine that has a frame rate, that has GPU acceleration, and therefore we use Three.js. It's made for all things 3D in the browser, and what we did is basically render all the points on a flat 2D space, fix the camera angle so that we just move the camera on one horizontal line, so that allows to zoom and pan, but uses the graphic card. So you will not even hear the ventilator going on my machine rendering 1 million points, and that's super impressive compared to the classical DOM approach. So here you see also in the little screenshot, you have one DOM element instead of 5 million. It's optimized by the browser for GPU, and yes, all the requirements that we wanted to build were easily possible by that.
Okay, now we are at 5 million points in the browser, which is already a lot. But if we look at similarities, which are very important in the analysis of this cell painting data, because oftentimes you have some regions in the space of that depicts the biology that you actually know, and you want to know which of your unknown treatments are actually close to that region. And if you look into pairwise similarities, then things start to get squared, right? So if I compare 5,000 conditions to 5,000 other conditions, suddenly I have 25 million data points because I have those pairwise similarities. So we thought, so what we had to do is come up both with a backend solution to compute these 25 million pairwise similarities on demand, because the groups are created actually by the user, and then also visualize them efficiently. And here we utilized Qdrant, which has a batch recommendation API. So basically, it comes from the similar items use case, right? You have a basket of or previously bought items, and someone takes one item, and from that stack of items, you want to get the most similar ones. That's the original use case. So what we did is we utilized this to basically go through the rows of our heat map and say, for each row, please, of all the column conditions, give me the most similar ones, all of them with the similarity score. And Qdrant does that very efficiently, and they realized it themselves, because when we built this workaround with the recommend API, we had to solve a few of challenges, and you see with our looping, we had to create these weird requests. By now, you can actually, there is a dedicated API called distance matrix API, where you can utilize Qdrant in that way to get these huge similarity matrices efficiently.
And to round this up, 25 million, we heard 5 million was the limitation of Three.js for a scatter plot. So how did we bring 25 million into the browser? Most of you might have more than millions in your pocket with your cameras. So nowadays, megapixels are obviously a good solution to render millions of pixels next to each other. So we thought to ourselves, how can we do that? Basically, what you've seen in the heat map is a fancy image zoom and pan functionality. We compute the squares on an off-screen canvas, so-called, and then simply drop one single image onto the canvas. And you might ask, okay, how do we do the interactivity? That's just computed based on the mouse position on the X and Y axis, which is 5,000 and 5,000, not 5,000 square. And this allowed us to easily bring 25 million into the browser, so by going another way around, which is totally sufficient for that use case. And to sum everything up, how do you actually render millions of data points? Computation, Matthias has told you visualization-specific backend interfaces and adapting the stack for the specific use case, but also maxing out the browser capabilities as far as possible, using as little DOM elements as possible, using GPU-accelerated technologies, basically the concepts of a game engine in the browser. And last but not least, filter and select. We are currently at the point where we have now maxed out the browser capabilities, but our stakeholders are already requesting more data also in the scatter plot. And then you can think of technologies, that, for example, like in Google Maps, you don't see all the streets when you look at a continent, right? So thinking about computing only what you need to see in that moment. And the concept here is called tiling, and that's what we're looking at currently to decide which points to render based on the zoom factor. That was it from our side. Looking forward to hear some questions, but thank you for listening.
Thanks.
Thanks a lot for your talk. Great talk and really impressive work you did there. We do have some questions. By the way, you can vote the questions if you want it to be featured. So the one with most votes is Did drop of the GraphQL validation cause any data-related problems with visualizations? Was the data quality high enough not to need the validation?
Yeah, it didn't cause any problem because we only did it for the output. So we have still full validation for all inputs, where we have mostly batch inputs, only very small inputs that actually are in real time. And so we are pretty sure that the data is consistent in our data storage, and so we just output it without validation.
Second question is, how long does it take from the time scientists to upload data and for the data to be ready to be interacted with? Was this a priority to keep low so that scientists can work with their data as soon as possible?
Yes, it was actually our key story internally. I think the reference was 4 weeks for analyzing what we refer to as a screen, so a bunch of experiments. And we wanted to get to start in the morning, so after image upload, I should say, start in the morning and come back after lunch and analyze it. Say you can do it the next morning by now, but that's where we are.
Okay, last question. Did you also try to build the front end with Python?
No, the stack for the front end is Next.js and React. There were some initial drafts, there are some libraries, but also here we run into all of these limitations with millions of data points. So most of I think Prosit, for example, would be one where you can render also simple graphs, but these are just freezing the browser. And building it entirely by ourselves, we also decided not to go with any libraries because we wanted so many custom functionalities in there that we wanted complete freedom. So all of what you see are handmade, built widgets in React and TypeScript and Next.js.
Okay, last question. We've got a new one. Why did you use Fast API? Which feature did you use?
Well, one reason is that it's quite common in our company, so there's a lot of knowledge about it. For Python backend development, we utilize the async functionality a lot, and it serves a great purpose. I'd rather question the choice of GraphQL than of Fast API, to be honest, in retrospect.
Great, so we don't have more time for questions. If you have any further questions, you can leave at the website or contact them.
Yeah, feel free to text us.
Yes, so thanks a lot.
Thank you.